Module 1, Part 4. In the Part 4 of the Module 1, we will discuss some of the incredible modern advancement of conventional fluorescence microscopic techniques and super resolution microscopy. Super resolution microscopy pushed the limit of light microscopy beyond its theoretical limit towards the resolution possible only through an electron microscope earlier. In the specimen that we observe under the microscope always have some thickness. However small it is, it could be 5 micron thick, which is literally a slice through a plane of a single cell, but there is a thickness at the microscopic level. Now, let's say you took that 5 micron slice of the cell, fixed, and labeled the sample with fluorophores and observing the sample with a conventional fluorescence microscopy. Now remember that the sample is 5 micron thick. You focused your microscope precisely at the 3 micron plane of the 5 micron thick sample. So only the sample at the 3 micron plane is perfectly in focus. But the 1 and the 2 micron level are above the 3 micron slice is out of focus. The same way, the 4 and 5 micron planes below the 3 micron level is out of focus as well. So when you are visualizing the 3 micron plane in perfect focus, out of focus light from the planes above and below the 3 micron plane level is also contributing to the image, and you get an overall blurry image, just as shown on the left side of the figure. Now, deconvolution microscopy uses a computational method to understand out-of-focus light, which is known as the calculation of point spread function in optical imaging. So after obtaining the initial image, the computational algorithm removed the fluorescence coming from the different planes. So how this microscope works to get a final sharp image? The microscope focuses individually in each plane and collect the optical data or the fluorescence from that plane only and cancels all the noise or the out of focus lights. So say first it focuses at the 1 micron level of the 5 micron slice of the sample that we were talking about and collects fluorescence data or optical information only from that plane, canceling data from 2, 3, 4, and 5 micron plane. Then the microscope focuses and collects data from the 2 micron and then 3 micron and then so on. Now finally, all these individual sharp images from each plane are merged together computationally using what is known as the deconvolution algorithm to get the crisp in-focus image like this one on the right, where you can clearly see the cytoskeletal components and the nucleus with great clarity. A modification of fluorescence microscopy is laser scanning confocal microscopy or just confocal microscopy. Previous discussion of microscopy, I talked about techniques required for sectioning thick specimens for the light to pass through, which resulted in 3D information being lost. Even if you are using a single cell with standard light or fluorescent microscopy, they look flat and you lose the 3D information. You really can see what is going on inside the cell, like where is the Golgi in relationship to the nucleus. That question may be hard to answer with standard light microscopy or fluorescence microscopy. Also, even when the thick specimen is sectioned for light microscopy, you still get the light from above and below the plane of focus, which results in a blurred image. And even sometimes sectioning doesn't help that completely. But confocal laser scanning microscopy allows you to focus on a specific plane within your cells or tissue specimen. The physics of this microscope allows you to reject the light that comes from out of focus areas. So you get an extremely clear image of your plane of focus. This is a variation on fluorescence microscopy and usually uses fluorescence optics. So here is a diagram showing the concept of laser scanning confocal microscopy. It is very simple and elegant. The major advantage is that you can use a thick specimen and you don't have to section them. So you can preserve the three-dimensional information and collect the 3D information on the sample. The confocal laser scanning microscopy uses a series of pinholes and mirrors to focus a laser light point by point onto your specimen and at the same time rejects out of focus light. The laser light, the blue line, passes through the pinhole, is deflected by the dichroic mirror onto the mirrors that scan the light. The light then passes through the objective lens onto the fluorescent specimen and excites the fluorophores or the fluorescent stain at a single point of focus. The fluorescent light from the specimen shown in green passes back through the objective lens, the dichroic mirror, and through another pinhole to be collected by the detector. You can see that the pinhole rejects the out of focus light. So here is another illustration. The pinhole focuses light on the specimen and the specimen fluoresces. As shown in the B, that light is emitted from the sample at the point of focus and easily passes through the confocal pinhole to be received by the detector and seen on the computer screen. But here is the beauty of the confocal microscopy. Any light that is not coming from the plane of focus, as in C, will be largely excluded, if not all. In a standard fluorescence microscope, there is no way to reject the light emitted by the specimen from areas out of the plane of focus. So here is an example. This is a sea urchin egg stained with a fluorescent antibody, a fluorophore for tubulin. You can see that the tubulin fibers, but they are blurry because you are also collecting light outside the plane of focus. And then here is the same specimen with the image taken on a confocal microscope. You can appreciate a much clearer image and the detail of the structure you are staining as the focus is on a single point. Depending upon the application, there are two modifications of confocal microscopes. The first one is known as the point scanning confocal microscope. The point scanning microscopes are used to image static or fixed samples that don't change over time. Now, let's first understand what the microscope is going to capture as an image. Now, say your sample that you are going to image, the image will be a 2D image. Think this way, that when we have a photograph of an individual, it is a 2D representation of an individual who has the third dimension or thickness as well, but that is not there in a photograph. So the same way here we are considering a two-dimensional image of your specimen. So there are always two axes, X and Y, for any two-dimensional object. As we are not considering its thickness or third dimension, the Z-axis is omitted. So the point scanning confocal microscope on the left, you can see there are two dichroic mirrors, one for the X dimension and another for the Y dimension. So these mirrors rock back and forth to precisely focus the laser to scan your image at a single point. So each of these single point will have a value on the X and Y axis. So basically, using these mirrors, the laser scans the image, say on the X axis, from all the way from the left to the right. When completed, it will shift one point down on the Y-axis and start from all the way from the left again, all the way to the right, just as shown in this schematic. So what we achieve from this? Remember that a single point being scanned by the laser only that point will emit the fluorescence, and which will pass through the dichroic mirror, and the data will be recorded. Except that one point that is being scanned receives the laser excitation for the emission of fluorescence. All other points are completely dark and there will be no background fluorescence. In addition, if any background out of focus light is emitted, the confocal pinhole will eliminate it. Now, thousands of these optical fluorescence data from each and every point that laser scans will be stored in the computer and will be merged all together to produce a crisp and sharp typical confocal image. The only drawback of this process is that it is time consuming, as the raster fashion laser scanning of the entire sample takes some time and can be done a fast dynamic event in a cell or a live cell. The spinning disk confocal microscope shown on the right is used to observe fast dynamic events in a cell, which the point scanning microscope can't capture. In the spinning disk confocal microscopy, the light path from the laser is spread to illuminate pinholes on the coupled spinning disk. The first consisting of micro lenses to focus the light on pinholes in the second disk. The excitation light or the laser then passes through the objective lens and provide the illumination of a number of spots in the specimen at the same time. This is the difference from the point scanning method, where only one point is illuminated at one time. The fluorescence emitted from all these points passes back through the objective lens and through the holes in the spinning disk and is then bounced off a dichroic mirror into the sensitive digital camera. The pinholes in the disk are arranged so that as it spins, it rapidly illuminates all part of the specimen several times. As the disk spins at about 3000 RPM, very fast dynamic events in the live cells can be recorded. So spinning disk confocal microscopy reveals dynamics in 6 frames of a movie of microtubules in a 2 rod-shaped cells expressing GFP tubulin. So take a look at the middle of the elongated bottom cell at the 0 second, and then toward the top of the cell. At 30 second, there is a small white structure formed on the upper side, and you can see that this microtubule is elongated over time. On the other hand, in the middle portion of the bottom cell, there is a tiny spicule-like projection, also grew rapidly and elongated over time. Conventional point scanning confocal microscopy have some limitations if the sample is thick, say a live animal. The reason being, the excitation wavelength, say on the left-hand side of a figure, is 488 nanometer, is used to excite the fluorophore, which is emitting 507 nanometer light. Remember that 488 nanometer excitation wavelength by itself is in the visible range and produce background or scattering, particularly in the thick samples over 1 millimeter. This issue is resolved by the 2-photon excitation microscopy. In 2-photon excitation microscopy, near infrared wavelength are used, which has much less energy. Remember that shorter wavelengths, higher is the energy, longer wavelengths, less is the energy. Just think that UV in the sunlight is of shorter wavelengths, less than 300 nanometer, can cause sunburn, but say blue light, say in the above example of 488 nanometer, will not cause a skin burn. Anyway, the 960 nanometer near infrared wavelength had significantly lower energy, so it is not possible for a single photon to excite a fluorophore to emit the emission spectrum. So in this 2-photon excitation microscopy, two photons are bombarded almost at the same time on the specimen so that their energy adds up to the double, and that is enough to emit the emission spectrum. So the right-hand side is a figure shows that two photons having half the energy of 488 nanometer add up to the same energy to emit the 507 nanometer wavelength. So what is the advantage? The advantage is the 960 nanometer near infrared wavelength is invisible as it is beyond the visible spectrum of light, and thus in a thick specimen it does not scatter the light as the 488 nanometer is visible wavelength does. This produces a sharp, crisp image in a thick specimen. So here is a comparison of the conventional confocal microscopy with two-photon excitation microscopy using a model. The model is a two glass cuvettes with some kind of fluorescent material in it. As you can see on the left, the excitation wavelength of 488 nanometer high energy is causing a large amount of background by producing a cone of excitation outside the focal plane. So that background will interfere with the emission spectrum from that single plane of focus, even in the presence of a pinhole of the confocal microscope, which is supposed to cut out of focus light. It is just a limitation of the instrument in case of a thick sample. But as you can see on the right, the 960 nanometer near infrared wavelength is beyond the visible spectrum with no background scattering of light. The advantage of the two photon over the conventional confocal microscopy is that this technique can focus the two photons much more precisely, which is a femtoliter volume, on a focal plane without almost undesirable excitation outside the focal plane. So the two-photon microscopy can be used to observe cells up to 1 millimeter deep within a living animal. This is known as the intravital imaging because it does not excite the fluorochromes outside the plane of focus. It is often used in a live brain imaging, as you can see in the test animal. Here you can see the nervous tissue in an intravital imaging of labeled neurons in a lobster. Total internal reflection fluorescence, or TIRF or TIRF microscopy, is restricted to a specific focal plane. This type of microscopy is great when the cells and the samples are attached or growing on a cover slip. In TIRF, only about 50 to 100 nanometer of the specimen adjacent to the cover slip is illuminated by directing the light in an angle at which most of it is reflected back from this glass water interface rather than passing through the specimen. That is why it is called a total internal reflection. Most of the light that falls onto the specimen goes back by reflection through the objective lens. TIRF microscopy generates a very small region of illumination called as evanescent wave. Now let's take a look at the difference between the regular epifluorescence, means above fluorescence microscope, with TIRF. Please note that the previous slide showed the excitation light coming through the objective and the emission fluorescence is going back through the objective from above the specimen. This picture is upside down or inverted in relation to the previous slide, where the excitation light is coming from the bottom of the specimen and not above. In reality, the microscope system can be set up in both ways depending upon application. Now let's see what is the difference. The leftmost side figure shows the epifluorescence, which is the regular fluorescence microscopy, where the whole thickness of the specimen is illuminated by the blue light, which is the excitation light. So all the fluorescent labeled vesicles will be excited and emit the green emission wavelength. Due to this, the depth information can't be collected, meaning you can't distinguish which vesicle is close to the cover slip and which is distant. all will appear as a two-dimensional image as if they are all at the same plane or depth. But in turf, the incident excitation light is directed to the specimen in such an angle that most of it is reflected back. Only an area very close to the cover slip, 50 to 100 nanometer, or the evanescent wave or field is illuminated, and only the vesicles in those areas will emit the emission spectra. The other vesicles will not contribute to the emission spectra as they will not be excited and excluded from the excitation illumination. On the right-hand side, you can see the cytoskeletal structure of microtubules are fluorescently labeled. The top panel shows the entire depth of the cell and shows all the microtubules spanning the entire depth of the cell. The middle slide shows the microtubule only closer to the top of the cell or the cover slip that are in the evanescent field. The structure below the evanescent field are excluded, and that's why the right side of the middle inset is darker. The right bottom inset was created by merging two falsely colored epi red and the turf green images to highlight the microtubules that are close to the cover slip, the green, and how the red distance microtubules were excluded in the turf image in the middle inset. Another technique that takes advantage of the fluorescence is the fluorescence recovery after photobleaching, or FRAP. FRAP can be used to track the mobility of a protein in real time. FRAP relies on the fact that fluorophores do not fluoresce forever. When you shine a laser light on a fluorophore at its excitation wavelength, it will emit the light for a given time, but not indefinitely, and will lose ability to emit light, and this is known as bleaching or photobleaching. So FRAP takes advantage of the fact that you can photobleach fluorophores or cause them to lose their ability to fluoresce. So let's take a look at this example. The left panel is the cell surface with the green ovals representing transmembrane proteins spanning the plasma membrane, and the right panel represents a single cell as viewed by the fluorescence microscopy. So you stain the protein of interest with a primary antibody or with a primary antibody and secondary fluorescent antibody, or label the protein with GFP or RFP red fluorescence proteins as used here. Somehow you make the protein fluoresce. Then you apply the excitation light to the RFP fluorophore. You focus the laser light on one small portion of the membrane and you photobleach out the fluorophore in that portion. So on the left panel, the fluorophores are no longer in the red asterisk, and on the right panel, it is represented by a white or non-fluorescent spot. And then you allow it to recover, turn off the laser light to that area. If the protein is motile and can move in the plasma membrane, it will move and will fill the area that was photobleached. You can quantitate this and get a real number as data. This is another example of fluorescence recovery after photobleaching. The cell surface membrane is made fluorescent by a fluorescent antibody or GFP or RFP tagging. See, a section is bleached, and during the recovery, if that protein is motile in the membrane, the fluorescence in that region will recover. There is a way to quantify the amount of fluorescence. You can collect the fluorescence intensity using arbitrary units and quantify the fluorescence from that spot. So you can see the fluorescence of that spot before it was bleached is emitting approximately 3000 fluorescence intensity units, and immediately after bleaching, the intensity drops to 1000 fluorescence intensity units, and then during recovery about 150 seconds, it rises to about 2000 fluorescence intensity, which is about 50% fluorescence in that area. So that suggests that this particular protein is 50% mobile on the plasma membrane, but 50% is immobilized by the cell in some way. Also note that after photobleaching, the fluorescence intensity did not drop to zero, but dropped from 3000 to about 1000 unit. This is a significant drop in intensity and represent photobleaching. In real experimental setup, even after complete photobleaching, there will be some residual fluorescence and scattering background fluorescence that can't be omitted from the detector. Hence, some fluorescence will be detected all the time. But this background will not take away the merit of this elegant experiment. Another very powerful fluorescence technique is known as the fluorescence resonance energy transfer, or FRET, also known as Forrester resonance energy transfer. This technique allows to determine if two macromolecules within the cell, such as a protein or nucleotide, are in close proximity to each other or they are physically interacting with each other. FRET does not rely on a single fluorophore, but a pair of fluorophore. One is the donor and another is an acceptor. This is a very special property of these pairs of fluorophores, and that is that the emission wavelength of the donor is in the range of the excitation wavelength of the acceptor. Here is a commonly used FRET pair, a cyan fluorescence protein CFP blue and a yellow fluorescence protein YFP. CFP is the donor and the YFP is the acceptor. The emission wavelength of the CFP is very close to the excitation wavelength of the YFP. So here is the principle of the FRET. If you have a component in your cell stained with CFP and a second component, say a protein, stained with YFP, and these two macromolecules are not near each other, you can apply a laser light in the excitation range of the CFP. It will emit in the blue range and nothing will happen to the YFP because they are too far apart for the CFP to influence the YFP. On the other hand, if these two fluorophores are close to each other, say the CFP labeled protein and the YFP labeled proteins are physically interacting, then the CFP is excited by its specific wavelength of light and emits light. Some of the light that is blue will be absorbed by the YFP. Since the emission wavelength of CFP is the excitation wavelength of YFP, YFP will be excited and emit the light in yellow range. So if you have interaction between the two fluorophores, you will see the yellow light, but if there isn't any interaction, you will only see the blue light. So what are the distances? They have to be very close. If the distance is greater than 10 nanometers or 100 angstrom apart, you will not get any energy transfer between the CFP molecule and the YFP molecule. But if they are 10 nanometers or less apart, then you will see the energy transfer or yellow light. We will discuss FRET throughout the course with specific examples. The advancement of super resolution microscopy pushed the limit of 0.2 micron optical limit of light microscope closer to the resolution that was only achievable by electron microscopes previously. Eric Bedzik, Stefan Hell, and Bill Morner won the Nobel Prize in Chemistry in 2014 for their contribution to the development of super-resolution microscopy. This clearly emphasized the immense impact of their contribution to the scientific community. Please note that super-resolution microscopy is a generic name and is not just one type of microscope or technique. Rather, there are several different types of system and techniques and used as per the need and application. The first breakthrough that sets super resolution microscopy apart that they can generate light microscope images with up to a nanometer resolution. New approaches to super resolution microscopy circumvent the light microscope theoretical limit of resolution or the ability to distinguish two close objects. In structured illumination microscopy, or SIM, the sample is illuminated in a pattern of light and dark stripes, and several images are taken as the illumination is rotated, generating interference patterns that can be mathematically reconstructed to generate a higher resolution image. This is only possible by the advancements of computational power and the ability to run very complex algorithms. So the theoretical limit of the optical resolution is not solved by the optics, rather solved by mathematics, digital image sampling capability, and computational power. The image on the right shows the similar fields of the nucleus, lamine in green and the DNA in magenta, imaged by the conventional point scanning confocal microscopy, and by SIM, which improves the resolution about twofold. In stimulated emission depletion microscopy, or STED, the sample is scanned as in point scanning microscopy with a very small light generated by an emission laser and confined by a donut-shaped stimulated emission depletion zone. The sample at the right shows the part of a cell stained for actin fibers after imaging by point scanning microscopy and by STED. In this technique, the depletion beam is controlled digitally to mask the laser point of excitation to an area much smaller than what can be achieved by a confocal microscope. So you can see the comparison on the right of a point scanning confocal image with a STED image, which can push the resolution to 30 nanometer range. Another type of super resolution microscopy is photoactivated localization microscopy, or PALM. In this technique, at a time, the excitation wavelength illuminates just a few random molecules of the fluorophore or the fluorescent protein, say in a cell. This way, thousands of images are taken and stacked upon each other. Then mathematically, using the Gaussian distribution, the exact middle point of each emission light point from a single fluorophore or protein molecule is calculated. See that in the Gaussian distribution, the highest point is the black bell-shaped curve, is the exact middle of the molecule that is emitting the emission spectrum or fluorescing upon excitation. In the Gaussian curve, the middle point has the highest value in terms of light intensity being emitted. As you move away from the middle point, the intensity decreases. This is obtained by mathematical calculation after sampling thousands of images. So the image is reconstructed digitally, where the optical information from the highest point of the Gaussian distribution curve is retained and all other are cancelled out or deleted. This technique provides a super resolution image, as you can see a comparison between a point scanning confocal versus a palm image of the cytoskeletal structures. Light sheet microscopy have medium to high resolution, but it is considered super resolution not for the image resolution or magnification, but for the speed of image capture of an extremely fast dynamic event in the living tissue and large samples like live embryos. The reason it is called a light sheet microscopy because a sheet of laser light is directed like a sheet covering a plane of a sample. You can think of it as a CT scan or MRI scan imaging of a patient where the sampling of the tissue is done in sectional planes. The same way, In this light sheet microscopy, a plane of the sample is illuminated, and each point on the plane can emit a fluorescence emission spectra to the detector. The plane of the laser illumination or light sheet is changed very rapidly through the sample, so the fluorescence emission spectra from each plane is collected to give an overall 3D image of a rapid dynamic event throughout the depth of the sample. As a comparison with the other conventional fluorescence microscopic techniques, in the wide field sample fluorescence microscope, the whole sample is excited by the excitation wavelength and it emits the emission wavelength to be detected. In the point scanning confocal microscopy, each point on the sample is excited individually, and the information of the emission light is also collected individually and later merged together to form the image. In the light sheet microscopy, as you can see, that a single thin plane of the sample is illuminated, and the detection of the emission light by the objective is an orthogonal plane, means it is 90 degrees. It is 90 degrees from the plane of illumination. This allows the different planes to be illuminated by the laser rapidly, and the emission light is detected by the objective only in that corresponding plane which is illuminated by this light sheet. And rapid collection of these images can produce a three-dimensional dynamic image. This concludes the part 4 of the module 1.